In this deep dive video, we will explore the structure of telomeres, delve into the essential telomerase enzyme, and uncover the replication process of telomere ends. Our focus will be on both leading and lagging strands, shedding light on how DNA replication naturally shortens telomeres on leading strands while extending or maintaining them on the lagging strands. This video will challenge the common perception by giving due attention to the often overlooked leading strands when it comes to telomere replication. Let's start with the fundamentals. Telomeres located at the ends of chromosomes have a distinct structure that sets them apart from the blunt ends. They exhibit an asymmetric extension with a partial double-stranded DNA configuration. The top strand typically has a lot of Gs, so we call it the G-rich strand. And the bottom one, then, is the C-rich strand. The single-stranded portion that dangles is called the G-rich tail, forming an overhang. In addition, telomeres exhibit a repetitive nature. The G-rich sequence, TTA-GGG, repeats numerous times in vertebrates, including humans, while protozoans have T-G instead. In humans, this short 6-base sequence is repeated over 1,000 times on both G-rich and C-rich strands. The G-rich overhang is about 60 to 200 nucleotides in humans, but it is much more shorter in yeast. This repetitive region is 5 to 15 kilobases in humans. And if you do the math, that is about 1,000 to 3,000 repeats of TTA-GGG. Now, let's add some character to this DNA. Telomere DNA comes with a set of special proteins that decorate the repetitive segment. Two homodimer proteins, TRF1 and TRF2, bind to the double-stranded portion of the DNA. These proteins have their own dedicated partners, RAP1 and TIN2. On the G-rich overhang, we encounter POT1, which happens to be a single-stranded DNA binding protein. And to connect POT1 with TRF, we have TPP1 protein. These 6 key proteins will play a major role throughout this video. It is important to note that telomeres don't exist in such an extended form. Instead, they adopt a capped conformation. What does that mean? The cap is a unique structure that hides the G-tail overhang in a specific manner. Structurally, the cap forms a very specific loop, where the G-tail overhang invades the double-stranded DNA and results in a distinct 3-way DNA structure. To complete the picture, let's add the decorating proteins. The elongated DNA loop is called the T-loop, meaning telomere loop. The T-loop can span several kilobases in length, and its formation and stability relies on 6 proteins that I mentioned. Collectively, these 6 proteins are known as the Shelterin complex. There are other notable proteins at the telomere cap as well, like the CST complex, consisting of CDC13, STN1, and TEN1. Now, let's zoom in further on this intriguing 3-way DNA structure. The DNA that is displaced by the G-tail forms a compact mini-loop, which we refer to as the D-loop. The length of this displacement loop matches the length of the G-tail, typically ranging from 60 to 200 nucleotides in humans. Logically, you cannot have D-loops longer than the length of the G-tail itself. This pretty much sums up the telomere cap. In case you are curious, here is an actual electron micrograph of the T-loop. You cannot see the D-loop because it is buried somewhere in this tiny spot here. And this concludes the explanation of the telomere structure. Now, let's take a brief tour of the telomerase enzyme, which is a ribonucleoprotein, meaning that it is composed of both RNA and protein. The RNA component, commonly known as telomerase RNA, tends to be rich in cytosines. On the other hand, the protein portion of the enzyme has a crucial subunit called TERT, which is a reverse transcriptase-like enzyme. Notably, the N-terminal region of TERT contains a binding domain responsible for binding the RNA-DNA hybrid, specifically of about 8 bases. This forms the active site of telomerase. Since telomerase is a reverse transcriptase, it uses RNA as a template to synthesize DNA. The template RNA used is the C-rich telomerase RNA. This also means that the enzyme can only produce G-rich DNA. The important segment to highlight within the telomerase RNA is this 10 nucleotide sequence. The full telomerase RNA is about 450 nucleotides long. This RNA is embedded within the TERT protein, forming what we refer to as the telomerase enzyme. Without getting ahead of ourselves, it is intriguing to observe that this telomerase RNA is complementary to the TTA-GGG repeat which we discussed earlier. This complementarity enables the extension of the repetitive telomeric sequences. We will revisit this concept later in our discussion. While we're on this subject, let's also explore the origin of the telomerase RNA. Interestingly, non-coding genes within the subtelomeric regions are responsible for producing this repetitive RNA. Through transcription, these RNAs are generated and referred to as the tera, or telomere repeat-containing RNA. Subsequently, they undergo processing and associate with the TERT protein, giving rise to the telomerase enzyme. Now let's put all this in context of DNA replication. Imagine a replication fork approaching the telomere. Here we encounter a challenge. The DNA to be copied is highly GC-rich, and the telomere DNA-binding proteins have induced topological changes, resulting in a looped DNA structure at the telomere. Also, the G-rich nature of this strand makes it prone to form secondary structures like G-quadruplexes. DNA polymerases cannot move on G-quads. These features of telomeres are obstacles for the incoming replication fork. To overcome the problem, the TERF proteins recruit specialized helicases from the RecQ family. These helicases work together with the CMG helicase, ensuring that it can smoothly unwind the telomere without any major pauses or disruptions. As a result, the T-loop with its complex structure opens up and the telomere linearizes into a simpler DNA structure, allowing DNA replication to occur at the end. Now that we've covered the fundamentals, let's discuss the end replication problem. It's important to address a common misconception that traditional textbooks often present, which can be misleading and confusing when it comes to telomere replication. Contrary to popular belief, telomere replication is not solely an issue of the lagging strand. I have an exclusive extended cut of this video available on my Patreon where I expand on this misconception. In any case, let's continue our discussion here. As we have discussed earlier, special RecQ helicases play a crucial role in unwinding telomeres to facilitate replication fork progression. The picture of the replication fork at the telomere looks something like this, with its lagging and leading strand synthesis. Now, here's the key point in telomere replication. Telomeres on lagging strands are extended, while telomere on leading strands are shortened. This challenges the traditional understanding that often focuses solely on the lagging strand. Understanding telomere replication on both strands is essential to grasp the complete picture. Let's begin by talking through the leading strand telomere replication, the often not discussed portion of telomere replication. So, if we were to replicate the leading strand, the daughter DNA strand would look something like this. To help us navigate through the concept, let's assign some random positional markers A, B, C, D, F, and G. In leading strand, the template is the C-rich strand, while the daughter DNA synthesized is the G-rich strand. It is important to note that daughter DNA synthesis continues right until the very end, until the polymerase and helicases falls off and dissociate from the DNA. As a result, a blunt end is formed. However, blunt ends cannot form the distinct T-loop structure observed in telomeres. This presents a challenge unique only to the leading strand. T-loops are typically formed by the G-tail overhang, which does not exist in this scenario. So, to restore a G-tail, a portion of C-rich strand at the 5' end on the bottom strand needs to be resected or removed. But how does this resection occur? Well, when the leading strand is synthesized, the TERF proteins and other components of the Shelterin complex assemble on the double-stranded DNA. The TERFs then recruit specialized DNA repair proteins, such as the MRX complex and specific 5' to 3' exonucleases, like the Apollo exo, to perform the resection of the 5' end. Other important proteins like the CSD complex and the single-strand DNA binding protein POT1 can also mediate this process. This restoration brings the telomere structure back to its original state, preparing it to fold into the T-loop structure. But if we pay attention to the positional markers, we notice that the E, F, and G positions on the original leading strand are now missing, since they were removed. So the telomere has shortened. Now, you could argue that the telomerase enzyme can utilize this recovered G tail to extend beyond position D and lengthen the telomerase and restore E, F, and G positions. However, this scenario does not occur due to the negative regulatory role of both POT1 protein and the CST complex on the telomerase enzyme. As a result, you cannot recover lost E, F, and G positions. So the leading strand is actually shortened during this process. This explanation covers a single round of DNA replication. In the next round of replication, the leading strand will further undergo resection, resulting in the loss of position, let's say position C. This modified leading strand can then proceed into yet another round of DNA replication, where a portion of the strand is resected once more. As a consequence, position B is lost in the bottom strand, leaving only position A. I hope you see the pattern in the leading strands. With each replication cycle, the leading strand is consistently and asymmetrically shortened. Alright, now we move on to the lagging strand telomere replication. When visualized, the synthesis of the lagging strand has its own distinct pattern of Okazaki fragments. To enhance our understanding, let me quickly decorate the DNA with telomere-associated proteins. Now, a couple of challenges arise during this replication process. First, the replication of D, E, and F positions remains incomplete, since there is no available template to generate an RNA primer for the formation of a new Okazaki fragment. Secondly, the RNA primers at the front of the Okazaki fragments are displaced by the advancing DNA polymerase. Unfortunately for the last Okazaki fragment, there is no polymerase to get rid of the RNA primer. Then how do you remove this RNA primer? In contrast to the leading strand, our attention now shifts to the G-rich template strand. The solution to our problems begin with TPP1 and POT1 proteins, which recruit the telomerase enzyme near the telomeres on the lagging strand. Let's redraw the telomere with the DNA sequence of the telomeric repeat to illustrate this process much more clearly. The telomerase enzyme utilizes the RNA component to bind to the edge of the G-rich strand. Using the same telomerase RNA segment, the enzyme synthesizes a complementary DNA. After completing this step, the telomerase enzyme translocates ahead, rebinds, and resumes DNA synthesis. Repeating this translocation and extension process multiple times leads to an increase in the DNA length. In humans, telomerase adds only up to 60 nucleotides of DNA to the lagging strands. Now, if we redraw the lagging strand, we observe that the single-stranded DNA template is extended. This extension allows a significant amount of POT1 protein to bind to this DNA. Recall that POT1 is a single-strand DNA binding protein. Interestingly, POT1 alone acts as a negative regulator of telomerase. However, when POT1 and TPP1 are together, they help recruit telomerase. It's worth noting that extension of telomerase activity is limited to approximately 60 nucleotides due to the POT1-mediated telomerase release. In addition to POT1, other proteins are recruited to the telomeres, particularly polymerase alpha and primase, along with the RNase H enzymes. As we discussed in the DNA replication elongation video, polymerase alpha and primase are capable of synthesizing both RNA and DNA. This helps to remove the RNA primer in the front, and RNase H can remove this particular primer. In addition to POT1, the recruitment of all these enzymes is also in a major way facilitated by the CST complex, which is typically available in the S phase and is associated with the TERF proteins. With the completion of this process, we can visualize the newly synthesized lagging strand telomere and decorate it with the telomeric proteins. It is important to note that the telomeric expansion is also limited by the expansion of the CST complex. Therefore, the lagging strand telomeres cannot just be indefinitely extended. These proteins, including telomerase, are regulated in a timely manner during the late S phase. This specific timing determines when and to what extent a telomere is processed. Alright, so we started with position A to F and added a short G to J stretch of DNA, approximately 60 nucleotides in length. This extension and processing of the lagging strand DNA occur during DNA replication. And now, the lagging strand telomere can fold into the T-loop structure. It is important to note that telomeres are not extended at each replication cycle. There are specific mechanisms in place to regulate the extension lengths or even shorten the lagging strand telomeres after they have been processed. Perhaps that topic deserves its own dedicated video.